All right. Don't worry about it. Thank you for the introduction, and thank you very much for the invitation. It's been a great meeting so far. So I'm going to talk about some work from my lab. So we are interested in developing methods to decode or to better understand transcriptional regulation. And so these are computational methods, okay? I don't do any experimental stuff in my lab. So let me also begin by thanking the two extremely talented women who did this work, and also the funding agencies. And so basically, Sneha was the one who started the project, and Anushwa then extended it to other kinds of data sets and extended the framework. Right? And a lot of the work was done at NCL, where I was before I joined ISER Pune. All right, so we are interested in understanding the architecture of regulatory regions, okay, which to a point where we should be able to predict what kind of an effect a mutation could have, right? So of course, we know now that regulatory regions come in many different flavors, right? And they play different roles depending on the presence or absence of protein DNA binding sites, right? And so if you were interested in looking for crucial mutations that could affect gene expression, then you should be looking at these regions, right, where the protein makes specific contact. Okay, so I'm only talking about specific protein DNA interactions, right? And of course, so we know also that these specific interactions are tiny. So a blue protein might bind this little string of DNA, right? And it also, a protein typically also allows for variations in the sense that just because it binds here at this specific string of 8 nucleotides, elsewhere it might bind to slightly different nucleotides, right? We know that, and which is why we often use this kind of a representation to denote a binding specificity. All right. I'll talk a little more about this representation in a bit, but the idea basically is that this helps you, this kind of a representation, a logo representation, helps you detect potential binding sites, right? So if you have that. The problem is you don't have such a logo or binding specificity for many transcription factors. And also, just because you have it does not mean that you will be able to detect binding sites with high specificity. You get lots of false positives because the binding site, the region that you're looking at is so tiny. So what has helped, of course, is advances in high throughput sequencing technologies, right? So now we can measure many different types of biochemical activities along the genome with fairly high high resolution. And typically when you get data from such experiments, the usual strategy is to do some sort of an average analysis, right? So we saw an example like this in the morning, where we had CTCF site, and it was the idea was that if you suspect CTCF sites to be positioning nucleosomes, you would look at all experimentally detected CTCF sites and look at the nucleosomes around them, also experimentally detected, and you would get these nice pictures showing that CTCF probably has a role in doing that. Except that if you look at it from the point of view of CTCF, it's doing many things. In some, maybe a majority of the regions where it's binding, it is doing that, but in other places it might be doing something very different. And the fact that we have the data probably suggests that we should look for those smaller possibilities, right? The other possibilities. Another example is something like this, which is very common, like this. We were looking at, let's say, ChIP-seq regions and say that a motif is overrepresented, which is also an average analysis, and I'll tell you in just a little bit. So let's look very quickly at protein DNA interactions, right? You get these peaks at the end of experiments like ChIP-seq or cut and run, so on. Right? And the idea is now, because you have done this experiment, you get these regions along the DNA, right? And the assumption being that all of them should contain a binding site for the protein that you're interested in or you have profiled. Right? So that brings us to a very old problem in computational biology, which is motif discovery. Right? And this is something that was formulated long ago when people were looking at promoters of co-expressed genes, okay? Where you were looking to find a binding site of proteins, of one protein. Essentially, you're saying that your regions are long, about 100 to 200 bases, but your binding site is tiny, hidden somewhere. You want to locate that. And simultaneously, you also want to find the logo. So you don't have the logo already, right? So that's why it's de novo motif discovery. So the logo actually comes from a position weight matrix, which is a probabilistic representation of the binding affinity of the protein. So you don't know these numbers. If you knew these numbers, you could find these. If you knew these guys, the green regions, you could find the motif. But you don't know either, right? So you formulate the problem in a probabilistic manner. So the idea is you want to find those regions. Simultaneously, you want to find the motif, right? And there are many methods that do this. The most popular one is meme, right? And there are many others too. Meme and Homer are the ones that are very popular currently. Okay. And you also know that many, you don't have just a single motif. You could have multiple motifs in your data. So what you would do, or what these methods do, is after you find the most enriched motif, you mask it out, you rerun the whole exercise, and you keep doing this until you're done with either some significance level or whatever the user-defined motifs are. Yeah? Okay. Okay, so the problem is that the results that you get from these methods are not always as straightforward. Okay? So many times, sometimes, no enriched motifs make sense, or the motif that you want, really, you know what you have profiled, is not at the top. Okay? And then you have to explain the other motifs in there. And also, this formulation does not really answer the question, how is the data really explained by all those motifs? Right? You're not really answering that question. So we asked a slightly different question. Why should you even expect a motif to be enriched in your data? So if you think about what you're profiling many different types of protein-DNA interactions. Right? A protein may bind as a monomer or a homodimer, heterodimer, indirectly. Right? When you're cross-linking, you get all sorts of interactions in your data set. So when your data itself is coming from a bunch of diverse complexes, right? Means that your DNA that you're getting from such experiments after sequencing also is fairly diverse, right? So we said that instead of looking for one most enriched motif, then the second most, let's try to split the whole data into these different binding modes, right? So here's the pictorial idea behind the approach. So we call the method diversity. It's, yeah. So the idea is if you knew beforehand that there were 3 different ways that a protein was binding, 3 reasons for a DNA region to be reported in your assay. So let's partition the data into 3 with one motif enriched in each. Yeah, so that would tell us the 3 different ways that it's binding. So now, the model changes a little bit. You're still identifying the location of those pieces, so that's the same. But instead of a single motif, you are learning 3 in one shot. Okay, so it's a little hard to imagine, but if you write it down in a probabilistic manner, it's fairly easy. So here you have your DNA sequence. This is the unknown piece, the little piece of binding site. This is the unknown location. This is the motif parameter. You don't know these, but if you did know what they were, and also the mode that it's taking between 1 and m, if you knew everything, you could compute the likelihood of the sequence of the data, right? So basically, you're saying that you just use a background model for the regions which are gray. For the pink, you use the mode that is in there. Right? So this is a likelihood. You can get a posterior with appropriate priors, and it kind of works quite well. Okay, so this is old, so I'm not going to talk much about this, but this kind of builds the story for the second part. Okay, I'll just very quickly talk about results for this method, so you just to see what you get at the end of applying diversity. Okay, so here's a protein called REST, R-E-S-T, that is supposed to repress neural genes in non-neuronal cell types, and we know it's binding site, right? And when we apply diversity to data set to a ChIP-seq data set for REST, we get these partitions. I don't know how well you can see it, but you can see that there are, and then they're aligned afterwards, right? The alignment happens later, after the fact. So you get the different modes, and you see that there are different, so this is the canonical one, and then there are bits and pieces where things are slightly different. Right? And you can also, now once you have the different partition, different modes, you can check what's going on at those different sites. Here I'm just plotting sequence conservation, and you can see that the sequence conservation is very different based on whether you have the full site or just the half site. We also looked at nucleosome occupancy, because REST is also supposed to get these nicely positioned nucleosomes. And here you can see that the ones where REST is actually binding as a, with both the sites, half sites, you get a much nicer nucleosome occupancy. Or you just have to interpret this as wherever there's a white, there is a nucleosome signal, right? So just take my word for it that it does look nicer for these guys. And the ones where you have a single, you just have one half site, you don't get a strong signal. So here's an example of, there are other section of your data that's doing something else. So in neurons, story is different. REST does not seem to be very enriched. This is your REST, and then there's lots of other binding sites in the whole data set. So the study also said that we could not find too many REST sites. But sure, you don't find REST sites, but you find other potential cofactors. We don't know. Okay, all right, so moving on. Anushka started looking at this, and she's saying what other kind of data can we use to get in this manner? Because there's just so many data sets out there where you're not thinking about explaining the whole data versus you're trying to look at enriched regions only. So one obvious place was to use it for ChIP-XO or ChIP-Nexus. So here it's very similar to ChIP-seq, except after cross-linking, you subject the sample to a lambda exonuclease. So that's going to chop up your DNA from one direction only. So what you get at the end of this exercise are these nice footprints, supposedly very close to the binding site. Yeah. So here's data on SOX2. Many of these proteins you've heard about. So this is SOX2 as a transcription factor, not the gene, not the genome locus. And you can see what you get in addition to, this is after running all your, after pre-processing the data and after doing the peak finding and everything. You're not doing the peak finding, right? So peak finding is doing all this. So finding the positive reads for you, the negative reads for you, using both of them, deciding what is a bound region, right? So now if you think about it, your data continues to have different modes. But now in addition to the different modes, you also have this other data, right, that tells you about reads. So you should be able to use those reads directly in the model. Okay, so what now that makes it is a slightly harder, problem, in the sense that you, in addition to having your DNA, you also have these two read data, which is in this case binary, but doesn't have to be binary. Yeah, so now, in addition to learning the motif, you're also learning the little signal around it. So it's all jointly learned with no other information except for the DNA and the two sets of reads. Okay, so this was our original data, and this is what we find. So you find, I don't know how well you see it. Yeah, so you see, this is what you expect, or this one, either of them. Here's when SOX2 is dimerizing, I guess, with OCT4. And these are those modes. These are modes where SOX2 is binding alone, but even within them, there's a different footprint for all of them. So one question could be maybe we're just overfitting, yeah? But we don't think that's the case because we did other validation studies where we used this information to build a proper mixture model and used it to classify, right? So we looked, this has much more information than just having two modes, which is what our competitor finds. So the Chex mix basically is the only other real method we see that is using the reads also to find the modes. And but what they do is they start off saying that let's have the top enriched sequences, run meme or motif discovery on that, use that as an initial step, and then refine those motifs. So which is why you don't get enough, as much diversity as we do. The other problem is because they're relying on enrichment only, they're able to explain, they're not able to explain the full data set. Okay, what about other types of experiments? Protein DNA interactions are a tad easier because you expect a single mode of interaction. But if you think about things like enhancers or open regions, right? So you have many assays that are giving you that, or even the nucleosome acetylation that we saw, right? That's also a mark that you're getting. All of them are inherently giving you DNA that comes from diverse regions and for diverse reasons, right? So if you're looking at open regions, they could be open because they're promoters, they're enhancers, whatever, right? Similarly, enhancers could be enhancers based on what aspect of enhancers you're assessing or measuring in your assay. Right? So a single motif is not going to explain because you have multiple motifs in each region. So we had to develop a newer program, which we call cis diversity. We started naming these way before diversity became like a thing. Okay, so we've had this name for a while. And well, cis was added later, but so all right, so what are the biology-driven assumptions? So we know that only a set of transcription factors would be active during the experiment, right? And sequence activity, we assume, is driven by a combination of motifs. So the regions that you're getting down are because of a combination of motifs, not a whole lot, not many motifs. And eventually, your experiment is reporting a mixture of sequences with many different motif combinations, but there should be enough instances of each combination to learn. So what we said was now, instead of partitioning your data, it's essentially clustering for those who are more computationally minded. It's like clustering, except that you don't know what your features are in your clustering, right? So you're learning those as well. So you're learning the motifs that are driving your clustering algorithm simultaneously. So if you think about it, what you have is at one level, you have motifs. Combinations of those motifs are making your modules, right? And every type of combination has many instances, many examples in your data set. And all you have are this set of examples together. You just don't have any of this. Okay? It's a little like, for those who've done some computer science, it's a little like topic modeling, where you have documents which you want to split into topics based on words. There you do know what the words are because you're dealing with English. Here you don't know what the words are, so you're doing a little more learning than what's in there. So there's a lot of unknowns which we tackle with probabilistic modeling. And very quickly, here is, this is what it does in simulated data. So we have our DNA. We plant 3 modules, which are a combination across 5 motifs. So it's all planted. And cis diversity is able to recover all of them. So you can just ignore this picture, but I'd like you to look at this guy here, which is the circle color and the size denote the same thing, which is the probability of seeing a motif in that module. Okay, so what we're saying is all our DNA sequences are split into 3 modules or 3 partitions, and each module is a combination of motifs. Everything is being learned. Okay, it does wonderfully on simulated data. So how does it do on real data? Right, so the first thing we went step by step. Let's try core promoters, where we know a little more about what to expect. And so this is in fly. It detects, so none of this is known, right? So it is after we get the motifs, we go back to the database and see which ones they match to. So it learns the motifs that are the usual suspects, along with some that don't have a name. We don't know what they are. And these are the partitions that it finds. Okay? And we also went and looked at the downstream properties of these modules. And we can see that they are kind of split very nicely into promoters which are broad and promoters which are narrow. So by narrow and broad, this is just whether the transcription begins in a narrow area or has a relatively promiscuous, broad transcription start site range. Yeah. So I don't think I have results on humans, but humans is a far more complicated situation. We get a lot of motifs and lots of different modules in there. Okay, so but since humans, I have a picture about humans in ATAC-seq. Okay, so these are ATAC-seq. This is the data about ATAC-seq in mouse tissues, 20 different mouse tissues. So because they're 20, along with ATAC-seq, they also have for every region, they have what they call an entropy of openness, in the sense how cell type specific is that region. And so I'm just showing you one picture from the paper, which talks about brown fat tissue. And these are the, you can just look at this picture where you can see the different modules that are detected. It automatically segregates modules into based on motifs which appear to be more brown fat specific. This is all de novo. And these guys here are more constitutively active promoter binding sites. So I'm going to move this. We also have done something with single cell. Let me just move on to something that might be a little more of interest to this crowd, which is about CTCF. We heard a lot about CTCF. And so what we would also like to do ideally is not to just restrict to ATAC or core promoters. We would also like to see whether we can extend this method to look at 3D chromatin also, right? So we don't, so if you look at TAD boundaries, unfortunately the resolution is a little low, so we get regions are very high. So this method, I don't know if you realize how many parameters it had, it's slow. It doesn't do great on large data sets. But then we also know that CTCF is involved, so there's no reason to not use cis diversity on ChIP-seq data, on ChIP-seq data, because there also you can expect modules. So we did that on CTCF in human ES cells, and it finds a lot of cofactors which make a lot of sense, a lot of other motifs that make sense, like OCT4 and all those guys. But in addition to that, it finds many variants of CTCF. Okay, and I don't have a picture right now. In the interest of time, I'm not showing you, but they seem to have different characteristics and different function. Not, I can't say function, but different characteristics in when you combine with other high throughput data. Right? So, and they're all generally conserved. Okay, so if you see the conservation mark, they're conserved. So it's not, it doesn't look like random. It looks functional. So this is, if you want to see more pretty pictures, you can talk to Samarth, who is somewhere there. And if you ask him nicely, he might show you some pictures from CTCF. Also, Rahul Siddharthan has been working on this with me. He's been a collaborator for many years, and now we're looking at aspects of CTCF in this manner. He also is doing neural networks, which I have not talked about at all here. All right, sorry, I'm already over time. In summary, I'd just like to say, neural networks or machine learning is often considered black box, but I think high throughput experiments and the data that you get from there is also like that, because there are many different reasons that you can get the data. And it's a good idea, I think, to have an open mind, and you need to go beyond enrichment to look for, beyond going and looking for a common sequence. Instead, look for maybe the higher hanging fruit, look for the different patterns in there. And sorry for having going over. Thank you.
Hello, yeah, thanks, Leelavati. Yeah, time for questions. Okay, there.
Thanks. So in the topic modeling, how did you decide the word length? Or is that something that just comes naturally out of your analysis?
It comes naturally. So that's also a parameter. So we start off with something. So I didn't say how we learn the parameters. It's through Gibbs sampling or MCMC.
Mm-hmm.
So at every point, it is looking at the space. So from, let's say you start off with a width of 8, right? It's going to look at whether 9 should be sampled or 7 should be sampled.
Okay.
So it takes steps. And it could go, so it comes naturally is the answer. It's not set in stone. It's not a parameter that's set.
But you just naturally arrived at
Yes,
kind
but
of
you need
a
a starting
normal
location.
sort of motif size for like a B-zip transcription factor?
Yes. Yes. But the data is good, right? I mean, it comes from the data. Yeah.
Okay.
Yeah.
M is equal to 3, which is kind of arbitrary, but
what
would be the M for some typical proteins that you?
No, so that would depend. So we use Bayesian model selection to decide what the M is. So we use different M's and then decide that this is a good one. But I don't remember any, I don't know if there is any.
No, but that would also have some biological meaning that a particular protein can piggyback with this many.
Possible. We have not looked at that in that aspect.
Yeah, so here, very nice talk. So in general, I'm curious, like something like what Ranjith is asking, but mostly like what kind of actual biological context can you incorporate? For instance, even the concentration of a transcription factor can vary. And if the concentration is very high, they can go and bind to even weak sites. So in general, when people do ChIP-seq and so on to extract this, like, do they actually?
It's in the native state. You get some steady state snapshot. Yes, but that's a good point. Single cell ChIP-seq is not there, but single cell attack-seq is there. So there is something where we can modify the modify the framework. We are working on that. Not there yet.
So I was wondering, because we get these multiple motifs, whether they scale with the binding of a protein at different sites, because in the ChIP-seq we don't get a uniform binding across different sites. So does it correlate with the kind of motif you get, or different types of motifs you get?
So you're asking whether the strength of the peak or the height of the peak correlates? Yes. I did not show you that, but yes. You do see, I mean, the usual suspect, the ones that are a direct binding site and a strong binding site does get a high peak, yes. And the indirect ones get a lower one. So that's why we infer it's indirect.
So if we scale or if we quartile the binding strengths into different quartiles of a ChIP-seq data, let's say we get 3 or 4 quartiles. So do they correlate with the number of motifs sort of you get?
I don't think I understand the question. So if you look at a bigger set of binding sites?
No, if we quartile, if we classify the binding strength of the binding, let's say into 3 or 4 major classes. So do we see this similar number of motifs which we get?
No, that might not, because it's dependent on the data set size also. So because we penalize, right? So as you increase your M, the number of modes, you are going to get nicer and nicer looking modes. Okay, so we have to penalize somewhere. So we have a penalty term. That's where the Bayesian model selection comes into play. But if you just split it and take the top quartile, right? One possibility is, because it's not as diverse, because it may not have indirect binding, you may not find those many modes. That's possible. But But in general, I mean, because the size also matters, if you have more binding sites, you might, it doesn't, the number of modes will not necessarily scale with the,
And just a quick, so for the binding mode, do you have to make hypothesis about the shape of the plus and minus strand profile?
No, so it
Or
finds it.
Okay, it finds it. Okay.
Okay, wait. I'll tell you what the hypothesis is. So here, right? So this model, right, where you have the two reads, right? So you see the purple and the red, right? So those purple and the reds, there is some starting hypothesis, but again, with Gibbs sampling, we vary it. So we learn the best. So sometimes that blue region, blue peak may be much further away than the immediate binding site, where you see something like here, but in certain cases, it may be closer. So that's why it is able to find, well, in this picture, it's not very clear. Those signals at varying distances, whatever explains the data the best.
Okay, so you use that profile to more or less give a function to the
No, the function. Okay, the function is all.
Or function, I mean monomer,
so
multimer, or
a Bernoulli distribution. So every column is separate.
yes.
It's a yes or no. So is there a high red, low red, high red, high red? So it'll learn those. So it'll learn that pattern, just like the motif. So motif is a pattern over ACGT. So the reads are a pattern over 0 and 1. Very similar. No. These are narrow peaks. This is post-processing. Oh no, but this is chip XO. XO will be narrower usually. Yeah.
Okay, I had one quick question. So your own data was showing that when you have two different transcription factors binding, the motif is slightly different or more broad. So I was wondering, in terms of biological context as well, you have now tissue types from which you have aligned data, which is attack, histones, and multiple transcription factors. So would those data sets be a very good starting point for you to sort of test in combinatorial fashion whether, wouldn't this be better if you start with a more
with a better
data set which has more annotations to it in terms of marks?
So I think we have to look at what question we are solving. So if the question is, explain to me about this experiment.
Right, yeah.
Then that's what we are trying to do. But if you're saying, explain to me about all this whole subset of experiments, then we could, the question changes a little bit. But then it should, depending on what your data is.
Yeah. Thanks. Okay, if there are no more questions, let's thank Leelavati and